Welcome everyone. We're going to begin. As you can see, this is a different format, but I'd like to welcome you to the TN2 webinar this time. Normally we would be in a very nice cozy room all together, but unfortunately in these strange times, now we're stuck online. So here on Zoom, but I think we have a very nice program for you. So a little bit about TN2. TN2 stands for Translational Neuroscience Network. And what we aim to do with these series of lectures is provide talks from fundamental science, but also clinical science, especially neuroscience. And then what we aim to do is translate between the different fields. We do annual conferences with alternating topics, and one year usually it's neurology and the other psychiatry. So this year is neurology with a specific theme, and the theme is neuroimaging. So what we aim to do in this webinar is provide you with 2 talks, but there are actually several webinars that are still coming, and I'll show you those at the end of the program. And here you can see the full coordinating committee, and I'd like to thank everyone already because it's been a lot of work to put this together. So here's the program. First, the introduction a little bit, then Martijn Van Der Hoevel on the human connectome, and then Laira Airas on PET imaging. And then afterwards, we'll come back with some closing remarks. So now I would like to introduce our first speaker. It's Professor Martijn Van Der Hoevel, who is a biomechanical engineer. And Martijn Van Der Hoevel is a world-renowned scientist in the field of connectomics. And connectomics, that means the study of networks, and this is especially true for brain networks. The reason why I thought it would be especially interesting to invite Martijn here is because he bridges different fields of science, and not just between the fundamental and clinical side, but also between the different fundamental sides. So between mathematics, engineering, and also neuroscience. So recently he's moved to the VU, and that is also to start up a specific interest in genetics, but also I think it's very interesting for our research group because I'm especially looking forward to working with you more closely. So welcome, Martijn.
Thanks for the fantastic introduction. I'm going to share my screen.
Oh, wait a second. Oh, not yet. No, we still have another poll specifically about you. So science has led to a good understanding of the workings of many organs of our body, like our lungs, liver, and kidneys. And it's catalyzed finding treatment strategies for diseases that affect these organs. Do you think the brain is different? So we wanted to start off the discussion a little bit to do a poll in this way. Aha. Oh. 1/3 and 2/3. Okay, okay. Well, it will be interesting to see if you can change the minds of the people who said no, or maybe even the people who voted yes.
Yeah. Who knows?
We'll see.
Who knows?
Before we move on to your talk, I'd also like to introduce Professor Laira Airas, the second speaker after you, just to get everyone involved in both speakers at the same time. And Professor Laira Airas is a neurologist and the first professor of neuroimmunology from Turku in Finland. And Professor Airas did her PhD on basic immunology and cellular neuroscience, but nowadays her main research focus lies on understanding pathological mechanisms in progressive MS. She was the first to apply PET imaging actually to study neuroinflammation. And I'm especially excited about this work because for TN2, this is a specific interest to bridge fields, and this is what she does best, I think. So welcome, Professor Airas. I know you won't give your lecture yet, but I think it's good to say hi to the audience beforehand.
Hello, I hope you can all hear me.
Yes, great.
Good.
So let's quickly do also the poll for your talk as well. There the question is the adaptive immune system responsible for all damage taking place in the MS brain? No. Most people say no. Okay. Well, we'll have to wait a little bit to see if you can change the audience's mind as well. But for now, I think it's time, Martijn to go ahead. I'll stop sharing my screen.
Okay. If all is well, you guys can see my my screen. Well, good morning, everyone. In particular, thanks to the organizing committee for well, giving me this honorable slot to talk a little bit about the research that we do. Menno already introduced me very kindly. My name is Martijn Van Der Hoevel. I am a computational neuroscientist with a strong interest in the brain, and in particular about how the brain is wired. I'm working at the fantastic CTG lab at the CNCR here in Amsterdam. And well, Menno already mentioned, it's a little bit different from that we normally would do, but that also gives me, I think, a great opportunity to welcome you in my home. And it also gives me the chance to talk a little bit about things that are normally at my desk. And one of these things that are on my working desk is this. Can you guys see this? It is a 3D print of a brain. And I'm very excited about this. And well, I can imagine that you say, well, I have one too. That's not super special, Martin. It's just a 3D print of the brain. We have these for quite a while now. But this one is actually quite special. And the interesting thing is that it's not of a human. Actually, it is an animal brain, a brain of a chimpanzee, to be more precise. But the interesting thing, it looks almost identical to that of the human brain, right? It is a little bit smaller, but I am sure that I could have fooled you and say that this is a human brain. It is not until I just pinpoint you to tiny areas where we can see tiny changes, tiny differences with a normal human brain, but otherwise it looks almost identical. And I find that really intriguing. I find that extremely intriguing that almost all of the Lego building blocks of our brain, the human brain, is apparently identical to that of other animals, but still we claim to have much more advanced cognitive abilities. Don't get me wrong, right? Chimpanzees are also extremely intelligent animals, but still we humans, we tend to claim that we are super special. But are we? And if the outer parts of our brain are so similar to that of other species, what about the inner parts? What about the connections? What about how the different Lego building blocks, how these different areas of our brain communicate to each other? Is that also the same? And if that's the same, then what does this tell us? Does this tell us that there might be underlying core principles of how our brain is connected, some general principles of brain connectivity? And if there are differences, then how do these differences relate to perhaps unique type of human brain functionality? So the connectome is a complete description of all of the connections that together build our brain. Like your genome is a full description of your genetic information, your connectome is a full description of all of your connections. And the field of connectomics is the field that studies this roadmap of connections. So the first goal of the field is to build as good as we can maps of the human connectome, to reconstruct as many connections as we can and as good as we can. And the second goal is to figure out how the layout, the topology, as we call this, of this network might shape brain functionality. So the core aim of our field is that we try to go beyond individual brain regions. We try to go beyond the hippocampus or the prefrontal cortex. And then we try to go beyond individual connections like the insonata fasciculus or corpus callosum or the arcuata fasciculus. But our aim is to take a look at the brain as this global complex network and then trying to figure out how the layout, how the connections are placed might be shaping our spectrum of brain functionality. So the connectome is a comprehensive map of all connections of a nervous system. Now the question I want to discuss with you this morning is, are there some underlying basic principles of wiring of the connectome? Are there some general underlying features that govern the complexity of the connectome? And second, then if these principles are there, then how do they shape our spectrum of behavioral capabilities, our motor behavior, our visual behavior, but also our cognition? And of course, in the end, if these principles are there, then might they also play a role in shaping brain disorders? So first step, of course, is to be able to measure the connectome. How can we reconstruct the different connections? How can we build a roadmap? Well, there are, of course, several, and actually there are many techniques to do so. We can do this at the neuronal level by means of electron microscopy, but at the more macro scale level, we can also do this, of course, with neuroimaging techniques. We can use techniques like diffusion-weighted MRI to aim to reconstruct the anatomical pathways of our brain, as you can see on the left here, and we can use techniques like EEG and MEG, and to some extent also resting state fMRI to aim to measure the functional connections between brain regions. And a step that we often take afterwards when we have reconstructed this network is that we aim to try to describe this network into general terminology. And one of these things that could help us with this is network science. So network science is a branch of math that makes formal description of complex systems. And we then aim, once we have made this description, aim to look at certain properties of these networks. Well, this might sound complex and complicated, but actually it isn't. And that's the beauty of it. Because we simply describe the brain as this simple collection of nodes, which will be our brain regions, and then the reconstructed connections, which could be the diffusion fiber tracts or the functional connections that we reconstruct by means of EEG or MEG or resting state fMRI as the edges. And as such, we can make a complete description of all brain regions and all of their connections. And once we have done this, right, once we have described such a network, we can then look for certain properties. We can look for whether there are some areas that might have more connections than others. And whether the highly connected areas would then potentially also be areas in the network, brain regions, that might have an important role in combining and integrating information. We can also look at how the network and the topology of the network, right, the layout of how the connections are placed between nodes might support efficient communication paths. But you can easily imagine that if some edges would be differently placed in the network, then this might either facilitate or perhaps hinder efficient communication. So there might be individual variation in connections, how they're placed within the network, and a certain level of communication efficiency or behavior or function, if you will, of that network. And so and so on and on. There are many different types of network properties that we can study and then link or trying to find a link or an association with all sorts of behavior. And indeed, over the years, many people have tried to start using these connectome tools to do so. People have suggested that the efficiency of communication within brain networks might be related to cognitive abilities. There's been many studies going on how perhaps brain development from the baby to adolescent to the adult might perhaps reshape brain connectivity and how that parallels development of certain, again, cognitive skills. And in particular, people have also started to use connectomics and network science to study brain disorders, both neurological and psychiatric disorders. And these studies tend to suggest that indeed the connectome and the wiring of the brain might play an important role in shaping not just one disorder, but actually a spectrum of disorders. And I in particular like to come back to this later on in my talk about the consequence of this, about the connectome to be involved, not particularly in one condition, but actually very likely in many, many conditions. So ourselves, when I was a student and a postdoc later on, I was particularly interested, and still am, in studying the relationship between the connectome and mental health disorders, in particular schizophrenia. And one of the studies and findings that we did was that connections particularly involved in integration and creating these short communication pathways in the brain might be particularly vulnerable in disorders of psychosis like schizophrenia and bipolar. So this suggests indeed that certain properties of the network might be related to certain type of distortions or dysfunction associated with brain disorders. All right, so let's get back to our question, our main question of this morning. What are the basic principles of wiring of the connectome? And how can these network tools help us with that? Well, one way to do that, one approach would be to not just study the human brain only, but also start examining the brains of other species. And a couple of years ago, Olaf Sporns at Bulmer and I, we coined a new term for this, which we called comparative connectomics. Right? Of course, we were extremely inspired by the field of comparative biology in this. And the field of comparative connectomics aims to compare connectome maps across different species to try to find overlapping features, because we argue that these might give us strong indications of these general organizational principles of the connectome, because they survive millions and millions of years of evolution. So there must be real fundamental parts of how connectivity is formed. And otherwise, what are the potential differences? And then what do these differences mean in terms of behavior of different and across different species? So let's first take a look at these overlapping features, right? Because again, this could give us an incredible boost into finding our basic principles of wiring, which we are this morning so interested in. Well, when we started to compare this wide range of species, right, all the way from simple worms to bird brains, to rodent brains, to monkey brains like the macaque, but later on also of great apes, of chimpanzees, and of course humans. In the end, we're also a primate. If we compare all of these connectome maps, two themes seem to be emerging. First, all of these networks, again, all the way from simple worm brains to human brains, they tend to have a strong, what we call modular organization. They have a strong tendency to be organized into local subnetworks that are pretty strongly densely connected within, but relatively loosely connected to the other modules. And a strong underlying drive for this is that there is a drive towards parsimonious wiring in nervous systems. And the reason for that is simple. Connectivity is expensive. If you want to build a connection and to maintain a connection, that takes a lot of neural resources. It takes metabolic energy. So if you want to build a connection, then it better be very useful. So overall, nervous systems, in the end, they want to keep the amount of wiring as limited as possible to minimize it. So how can you do that? Well, if you want to cramp as many connections into a limited amount of space, because in the end, our brain, right, it has to fit within our skull. That's the limitation. It's the physical limitation that our brain has. Then if you prefer short-range connections, then you can cramp in more connections in the same amount of space. Now, these short connections, they form local segregated communities. And interestingly, studies have suggested that these communities, these wiring anatomical connectivity groups, they tend to strongly overlap to known functional systems, like the visual system or the auditory system or higher order cognitive systems. So the strong drive to minimize wiring might facilitate a segregation of information within these networks. But this drive towards this high community formation within networks cannot explain the entire level of complexity. So there is also a second theme that is very apparent, again, across all the way from the worm brain to the mouse brain to the human brain. And that is that all of these networks also tend to show a high level of investment in features that might bring topological integration. And these often are very expensive features of neuronal organization. Things like search communication paths, or for example, the existence of hubs within networks, right? Some areas that have many more connections than others, and as such might form a sort of integration-rich club within the overall system. So two themes, right? On one end, the drive to minimize wiring and which facilitates segregation of information within the network, beneficial for specialized brain functionality like visual, auditory processing, but also working memory, etc. And then the other end, a drive to form integration, because integration might be beneficial for higher order performance of the system as a whole. Now, it's interesting to realize, perhaps, that these two features, these two themes, are not independent of each other. Actually, they're more opposing forces. Because if you want to minimize wiring cost and go into a very strong modular type of network on this end of the spectrum, well, that means you don't have much integration. And the other end is also true. If you want to have a network with a high level of integration, well, then, well, you need to have a lot of investment in and invest a lot of neural resources in specific type of features. So in the end, it is argued that the connectome as a whole, the network, the organization, the topology of the network as a whole might be a trade-off. It is a balance between, on one end, to obtain maximal integration, but on the other end, try to minimize wiring cost and get segregation and specialization of information. So the connectome across many species two basic principles of wiring might indeed be this drive towards integration, and again, similarly, this drive to modular structure. Two principles of wiring that might govern the global organization of the human brain network. So mostly now we talked about overlap across species, right? But in a similar way, I promised you that comparative connectomics is also about searching for variation. And well, in the sake of time, I won't spend too much time on this, unfortunately, because I'm really fascinated by this. And because in particular, is the variation in connectome structure potentially also a foundation for variation in behavior? Not just, well, between individuals, but also, of course, between species. And I think both topics are extremely interesting. And we've already seen that within a population, within humans, variation in connectome structure might indeed be related to certain aspects of variation in cognitive abilities, for example. Now, across species, there isn't that much studies about this yet, but there are two, I think, in my opinion, brilliant studies that I urge you to start looking into if you're interested, as I am, as much in this topic. So one paper is a paper by Boombager and colleagues. Again, in the sake of time, I won't spend too much time on this, but it is a brilliant paper on comparison of the nervous system at the neuronal level between two closely related worm species. And they basically show that how neurons can connect to each other might shape different types of, in this case, eating behavior of the two worms. Right? One is a vegetarian and the other is a meat eater. So go read this paper if you're interested in this topic. I'm not a co-author, I don't have any stocks in this, but it's just a brilliant paper. Now, there's also more evidence for variation in connectivity to relate to variation in behavior more closer to home. And that is by the work of fantastic work by Jim Reiling and Todd Preuss from Emory University. So they, for example, showed that connectivity between language areas like Broca and Wernicke in the human brain is much stronger in the human brain compared to that of chimpanzees and macaque. Again, suggestive of certain investment, evolutionary investment in certain aspects of brain connectivity to potentially indeed relate to the development of unique type of features. So we got really interested in this a couple of years ago, and I was lucky enough to start working with my good collaborators, Jim and Todd, on this. And we started a project in which we said, if we can reconstruct a human connectome, can we perhaps also reconstruct the chimpanzee connectome, right? We know that we can measure the cortex, but can we also measure the inner parts? Can we measure the wiring? And this is fantastic work performed by Jake Dash in the lab. And interestingly, when we reconstructed the chimpanzee and the human connectome, indeed, we found huge overlap. Right? Many of the connections are simply shared, which again supports the idea that many of the underlying principles of wiring are shared from humans to other species. And they're very core elemental features of how connectivity is formed. But we also find tiny differences, some potentially human unique type of connections. And these human unique connections we mostly found to be existing between multimodal association areas. Suggestive that there might indeed be an investment of resources, particularly in features that might bring topological integration, beneficial for higher order cognitive abilities. And even more recently, this is work by another student in the lab, Yongbin Wei. Well, actually, he's not a student anymore. He graduated last week. That's why he has such a big smile on his face on this photo. But basically, he showed that in particular networks like the deformal network and frontal-parietal network are mostly expanded in the human brain compared to that of our evolutionary cousin, and that human accelerated genes that we know to be important for the evolution of us humans and the diversions from chimpanzees were particularly expressed within these higher order cognitive networks. All right, so let's end this intermezzo. And let me try to wrap up. But trust me, I have many more to discuss about this. So if you're interested in this, then make sure to give me a call. So two principles of wiring, right? On the one end is drive towards local connected communities, and at the other end, a drive towards topological integration. Now let's switch to disease, because if these principles of wirings are so crucial for our brain organization, and brain behavior, and potentially also human unique type of functionality, then it must almost be inevitably that they also shape and play a role in brain disorders. And indeed, when we start digging into literature, this seems to be the case, right? Hubs, as I mentioned, have been reported to be playing an important role in things like schizophrenia. But many others have also reported that they might also play an effect in other disorders like Alzheimer's or schizophrenia. So this suggests that indeed hubs might play an important role in the brain pathology in a wide range of disorders. And at the same end, also modularity has been suggested to be a key player in shaping pathological patterns, not just in one disorder, but a range of disorders. So this we started to examine by the work of Simon de Lange, another student in the lab, in which we simply said, well, can we basically compare connectome alterations, not just in one disorder, which we have been doing for schizophrenia and autism and OCD and MDD, but every time one disorder at a time. But we said, can we also make a comparison? Can we do some sort of a comparative disease type of connectomics? And indeed, what we found is that if we start looking at the pathological patterns of this wide range of connections across this wide range of disorders, and we showed that indeed connections that are important for global integration in the brain might be particularly vulnerable, or at least involved across this, not just one disorder, but much more of a wide range of disorders. So what's the next step? Now that we hopefully have found a first glimpse of these basic principles, then what is the next step to start examining these principles? Well, one of the things that we particularly want to do is we want to dive into the origin of these connectivity principles. Where do they come from? And how can they become disrupted? And for this, we have to realize, of course, that the human brain works at many different levels at the same time. In that sense, it's a multi-skill type of network. And so far in my talk, I focused on the connectivity level, at the system level. But of course, we have to realize that, again, the brain operates at many different levels, right? From genetic information to neurons, to connectivity to systems, and together they form the spectrum of behavior, which then forms the spectrum of mental disorders. So we are very interested in trying to bridge this system level approach with these other levels. And we aim to do that to get a more complete view, a multi-skill view of the emergence of these brain disorders. So how we are aiming to do that? Well, we want to connect how cellular changes perhaps relate to macroscale changes in disorders. We also want to look for how the expression of genes at certain areas of the brain, mostly spatial transcriptomic data, how that might relate to the organization and the formation and the development of connectivity. And might it indeed be the case then that genes related to certain specific brain disorders, are they perhaps overexpressed or underexpressed in particular areas of the brain? And how does that relate to the pathology pattern that we see in the connectome? And of course, we're aiming to make advances of the fantastic new technology being developed in terms of GWAS studies, right? In which we are able to map genetic variation to variation in phenotypes. And in a similar way, we can look for whether there's individual variation in brain networks and how does this relate to genetic information. And it too, does a genetic vulnerability, does that coexist with a connectome vulnerability of brain disorders? So to summarize, right, I hope that I convinced you that the brain is a network. Well, that wasn't a hard job to do, I think, but that there are potentially underlying core principles of how certain brain areas are connected and what the consequences are of these general rules of connectivity. And that we have our general aim for the future to start figuring out potential cross-related species, but also how genetic and environmental diversity and variation might shape in turn variation in brain function and relationships across disorders. So many people to thank. If your name is not on this, rest assured, I'm still very thankful for your collaboration and all the work that we've been doing. Well, I'm very much looking forward to our discussion.
Thank you very much, Martijn, for this fascinating talk. What we wanted to do now is to give the floor first to someone to ask you a question through video. So if you stop sharing your screen, you guys become larger.
Sorry, you want me to? Stop sharing. Yeah. So
yeah. That's right.
Yeah.
Okay, great. So I'd like to introduce.
Slides are interested in my face, but
yeah. I'd like to introduce Linda Dow, who's an associate professor of the Department of Anatomy and Neuroscience of the Amsterdam UMC, also a fellow network enthusiast, which is why we thought it would be great for you guys to have a small discussion. So go ahead.
Yeah, thanks, Martijn, for an excellent talk. Very interesting to again see your thoughts. And as Menno already indicated, I'm of course also a network enthusiast. And I see in the Q&A that there are many questions about networks. So more detailed questions. And I think those will be very interesting to answer after this. So I want to briefly start by maybe challenging you a little bit about clinical translation, because we are here at the translational conference. So I was thinking, I have two questions and you may decide which way to go based on whether you like to focus on the positive or on the negative. So my first positive sort of question is, if we had to bet on one disease or one network modality, or pick one thing that will be the key towards translation, what would it be? So that's kind of where do we go? Or what do you think, if you want to focus a little bit more on the problems, like what is the major hurdle that will stop these wonderful insights from making it to clinical translation?
Yeah, so I'm going to try to answer them in just in one answer. I don't think that there's one particular disorder that is more strongly connected to connectivity than others. I generally start to get more interested in cross-disorder effects myself, and that might manifest itself into various different types of symptoms, right? Which we then tend to classify into disorders. A little bit in a similar way of people start to think about genetics, right? Here too, we talk about vulnerability much more than specific genes to be related to specific type of disorders. Well, of course, that also happens, and we call it, you do have conditions where there is a single genetic malfunction, and then you end up with a specific type of condition. But here, we're talking about much more complex disorders, mental disorders and neurological disorders. So I tend to more and more think in general vulnerability factors. So what are the biggest hurdles? I would just like to call them challenges, things that we have to face. I think there are many things we don't understand. Some of them actually were also, I think, postulated already in the or asked in the questions. What about structure and function? How can we start combining them? And I definitely think that is, of course, an important step that we have to take. In the end, the brain isn't only anatomy. The brain isn't only function. So we have to understand the relationships. And personally, I think to address that in a much more broader sense, that as I mentioned, in terms of multi-skill neuroscience, I don't think we should only try to measure structure and function and try to link that together, but that we have to start realizing and invest, I think, in how does the tiniest building block of our brain, the neurons, relate to the macro-skill systems. I think it's becoming more and more evident that it's not just if we just pile up a lot of neurons together that we end up with the brain. At each level, there is a certain level of complexity that we have a hard time yet to understand yet. And that each one of these levels in some way have to be bridged to the other levels.
Yeah. Yeah. So
more
that
for me would be the biggest opportunity to dive into.
Nice. Thanks a lot. Menno, maybe you want to go to the other questions.
I can also see them.
You're muted. Oh, okay.
Okay. Let's see, which one shall we do? Just reading quickly. So here's one from Jacqueline van Vierbergen. Under the assumption that brain connectomics is based on the principles of criticality, do you think that neurological disorders actively shift the brain to subcritical or supercritical states? Or do you think that the window size for criticality has become more narrow and will therefore by default fall into a non-optimal critical state?
Yeah, it's a fantastic theory that the brain is that sense. If it's a complex system, is likely also a critical system, although it is a little bit of shifting of terminology, I think. But that's indeed actually a theory that we proposed within the spectrum I showed of the trade-off. We more and more start to think that the brain actually tips the trade-off into a suboptimal domain and that this moving away of the connectome into this new shape, that this might be shared across disorders. So that might be an underlying shared feature of how the connectome plays a role in a wide range of brain disorders. Well, it almost gets much more mathematical and philosophical. So I'm always a little bit hesitant to push these things to forwards in these talks. It is a hobby of mine to think about these things, but I like Linda's comment to say, what will be the next step to make this into a translational phase? And I always like to, again, do both and try to see how we can make the translation. But I think the critical theory in general, or the theory of critical organization, it is a theory. So it is extremely important to start proving this, because that's the first step in making the translation in the end. And to me, and I think also to Linda and many people in Amsterdam, that's of course is our end goal, right? Not just to show that the model works, but also to, in the end, to do our tiny little effort and step to help the bigger picture.
Yeah, great. Let me see another one. Here's Sophie van der Sluis. Would it make sense to focus studies on brain areas that are dysfunctional in brain disorders to those that show differences between humans and chimpanzees? Do you think there's a relationship between?
Yeah, absolutely. Well, absolutely. Well, Sophie knows my hobbies, and Sophie is a close, was my colleague. So absolutely. And I think there is one particular. There was also some few questions about specificity, whether I could refer to some specific type of disorders. And one of these disorders is schizophrenia, and it has long been suggested to be actually only present in humans, to be a relatively human unique type of brain condition. It is not present in chimpanzees in the wild or in other species. In contrast to, for example, other types of disorders like depression or OCD, there are some symptomatology that seems to reflect some presence in the animal kingdom. And indeed, when we started to look into comparing connectome changes in schizophrenia compared to evolutionary changes, we indeed tend to see a strong relationship. And this has been connectome evidence, if you will, of a long-lasting theory that indeed schizophrenia might be related to an evolutionary change of the brain. And in general, I think there is a general interest in thoughts of that our expanding brain in terms of evolution. That's a fantastic thing. It's going to give us a lot of cognitive capabilities and richness, if you will, but that there might also be a negative side effect to this, and that we humans might be more vulnerable to a wider range of brain disorders. So that's definitely something that we are playing around with. And
yeah, I think also that really highlights the feeling we're trying to portray with TN2, that even though you have very fundamental insights in the evolution of the brain, they are still clinically relevant. So this translation is, I think, very important to explore further. With that, I would like to thank you for now. Stick around until after the next talk. But I would now like to give the floor to Professor Laira Airas, as I said, Professor of Neuroimmunology, who will enlighten us on multiple sclerosis and especially the new and exciting field of PET imaging.
Okay, thank you very much. So good afternoon all, and hello from Finland. It's a great pleasure to share with you some of our recent work on PET imaging of activated microglia in MS brain. And I thank the organizers for this kind invitation. I must say that my presentation will be very much more down to earth than the previous fascinating presentation on the connectome. So I'm a clinical neurologist. I look after MS patients, but also we are very much trying to figure out what's happening in the brain of MS patients when they turn into the progressive phase of the disease. So MS is the most common neurologic disease of young adults. It's considered to be an autoimmune disease, and it usually begins with episodic attacks of neurological symptoms and will enter a progressive phase some years later. So the onset of the disease is on average at age 30, and the prevalence in Europe is about 1 in 1,000. We don't know exactly what causes MS, but complex interaction between a genetic predisposition and an environmental trigger is thought to initiate the autoimmune inflammatory attacks. So these are the hallmarks of MS pathology. So there is an initial autoimmune activation of the adaptive immune system, and this leads to focal demyelination, to innate immune cell activation within the brain, and then to axonal and neuronal loss, and to astrocytic gliosis. So as I said already earlier, so MS patients are 30 years old on average when they get the diagnosis of relapsing remitting MS. And more than 60% of the patients convert to a secondary progressive disease on average at the age of 45. So we have very good treatments for the relapsing disease, but not much to offer to patients as they convert to secondary progression. And the lack of efficient treatments for progressive MS is considered the greatest unmet need in the field of MS at the moment. And there are many ongoing efforts to try to understand what's going on pathologically within the central nervous system to help develop treatments for this disease type. So the relapsing remitting MS is associated with abundant trafficking of immune cells from the periphery into the central nervous system. And this results with focal inflammatory lesions, such as shown here in the MRI and around this blood vessel here under the microscope. There's this really thick lymphocyte infiltration. And the acute lesions are enhanced with gadolinium. And when the disease turns into the progressive phase, the pathology changes. It looks a bit different. So there's probably still some inflammatory cell trafficking from the periphery, but this is much less than in the relapsing remitting disease. And this is seen as fewer gadolinium enhancing lesions in the MRI. But instead, the inflammation becomes trapped within the central nervous system behind the blood-brain barrier. So the pathology is spread also outside the focal lesions, and there is more microglial activation and more oxidative stress and mitochondrial dysfunction. And all of this contributes to axonal damage, which then leads to brain atrophy and disability. And this is really the end result that we would like to avoid. In some cases, we just see this very severe brain atrophy, luckily not in all. So if we are thinking about the lesions in progressive MS, so we see these typical chronic lesions in the T1 weighted MRI images. So these chronic lesions, they don't enhance with gadolinium. And in this conventional MRI, they look identical to each other. But we, however, know from neuropathology that there are varying pathologies behind these T1 chronic lesions. So in the histological analysis, you see lesions with a rim of activated microglia, like here, and these can be called chronic active lesions. And then we have lesions with a much less microglial activation at the lesion edge, and these are called chronic inactive lesions. But then there's also activated microglial cells in areas of the brain which look perfectly normal in the conventional MRI in the normal appearing white matter. So microglia belong to the innate immune system of the central nervous system. And they are small but important cells which are normally needed to keep up the tissue homeostasis in healthy brain. But in chronic neurodegenerative conditions, they can become overactivated, and this can create a state of uncontrolled inflammation within the central nervous system, and this contributes to axonal damage and neurodegeneration and disease progression. So there's sort of this vicious circle of pro-inflammatory activity. So in neuroinflammatory and neurodegenerative disease, microglia acquire the pro-inflammatory phenotype. So they are too hot. They secrete pro-inflammatory cytokines, chemokines, and free oxygen radicals, and also promote increased release of glutamate. So let's consider the evolution of an MS lesion. So in an acute lesion, the lesion core is filled with macrophages which clean up the myelin debris. So this is required for efficient remyelination, but with time, the lesions evolve into chronic active lesion. And here, the edge of the lesion here is formed by activated microglial cells and macrophages, which have a pro-inflammatory phenotype. And at this stage, the core of the lesion becomes acellular with just a gliotic scar. And some of these lesions progress into chronic inactive lesions, which don't have the activated microglia at the lesion edge anymore. So the inflammation dies down and the edema resolves. So particularly the chronic active or the smoldering lesions are considered harmful in terms of neural damage and disease progression. And conventional MRI, which is the most commonly used imaging technique in MS field, it cannot be used to detect the activated microglia. And the MRI metrics don't correlate either very well with clinical signs of disease progression, so better techniques are needed. So the activated microglial cells upregulate an 18 kilodalton translocator protein, or TSPO, and this is used as a target for PET imaging of neuroinflammation. So these figures are from our own work. So here in this MRI image, you can see two identical looking chronic lesions, but in the TSPO PET image next to it, only one of the lesions, this upper one here, takes up the radioligand, and this demonstrates that this is a chronic active lesion, whereas the other one is a chronic inactive lesions. And the figures in the bottom also show examples of TSPO PET images obtained with a PK11195 radioligand. We have shown that TSPO PET measurable microglial activation increases in the normal appearing white matter and also in the thalamus as MS disease advances, and there is significantly increased radioligand binding in the secondary progressive patients compared to relapsing remitting patients or controls. Okay, so here's the timeline of TSPO binding radioligand development. The first TSPO binding radioligands were developed already in the 80s. And one of these, the PK11195, is still in use. So it's a carbon-11 labeled ligand, and it has been used in most PET studies performed in MS. We have also been using mainly this ligand in our studies. We use high resolution scanner, which has a resolution of 2.5 millimeters, and we do a 60-minute dynamic scan. So the PK11195 radioligand has its shortcomings, such as a low signal to noise ratio and a lowish penetration into the central nervous system. And this has led to attempts to develop better performing TSPO ligands. And in the top are radioligands, which are already in the clinical phase of development or in clinical use, and the ligands displayed in the bottom are still in earlier development. So the new ligands also have their problems, such as the variation in binding potential between individuals due to polymorphisms in the TSPO gene. And TSPO imaging is far from clean, also in a sense that also other cell types apart from microglia express the TSPO molecules, such as endothelial cells, macrophages, and reactive astrocytes. Really new better ligands are needed for improved specificity, but it has been surprisingly difficult and slow to get new tools for imaging of neuroinflammation. So the PK11195 ligand has its shortcomings, but it also has significant advantages over the second generation ligands. The analysis methods are quite well developed, and they give consistent results, and no arterial sampling or genotyping of the study subjects is required. So in our studies, we use the Turkheimer method, the supervised cluster algorithm for reference region extraction, which is necessary as there is no normal anatomical reference region in the MS brain. By now, there is over 20 years of experience of TSPO imaging of MS brain. The earliest studies demonstrated the very strong radioligand binding in the acute lesions, but they also showed the correlation between the increased binding and disease duration. To us, the fact that you can visualize TSPO binding in acute lesions is not so interesting, since this is something one can easily detect also with conventional MRI and gadolinium enhancement. But when you can use PET for detecting pathology that conventional MRI cannot demonstrate, so we think that's when it gets more interesting and more important. So this is an image from our own work showing at group level the areas in the normal appearing white matter where the PK11195 binding is increased in SPMS patients compared to controls. And here is a demonstration of a chronic T1 lesion with visualization of this PK binding at the plaque edge with a 3D surface plot here. So here we are sort of looking into the lesion. So the lesion core doesn't bind the radioligand, but at the edge of the lesion you have this like a mountain range showing the increased ligand binding. So at the moment we have imaged over 100 patients using TSPO PET, 100 MS patients. And here is shown results from 91 patients with PK 11195 imaging in relation to disability. So this higher distribution volume ratio associates with more disability, both in the normal appearing white matter and at the perilesional areas. So more disability, more microglial activation. Now we have recently published a paper demonstrating that DTI MRI measures reporting on neuroaxonal damage in the normal appearing white matter also associate with increased microglial activation. So there is co-localization of microglial activation and damage. And here, this is from our most recent publication from a few weeks ago, which really gives proof of concept that TSPO PET measurable innate immune cell activation is detrimental for MS patients, as those patients with more microglial activation progressed more rapidly during the following 4 years. So this raises some questions like, could this harmful microglial activation be targeted therapeutically? Or does the modulation of microglial activation influence the development of neurodegeneration? In other words, can the smoldering fire be extinguished? And could this be measured using TSPO PET? So we have done some longitudinal PET imaging and shown that in untreated patients, microglial activation increases over 1 year of follow-up, here shown both in the normal appearing white matter and around the lesions. But if the patients were treated with natalizumab, there was actually a decrease in PET measurable microglial activation. So in other words, with this, we sort of indicate that the smoldering fire can be extinguished. So of course, natalizumab is a monoclonal antibody, and it doesn't really penetrate into the central nervous system, but it very efficiently blocks the entry of the adaptive immune cells from the periphery into the central nervous system. And so our thinking is that natalizumab just calms down the inflammation very efficiently in the brain. And then this also sort of leads secondarily to reduced activation of the microglial cells. So, so far we have published a number of reviews also on PET imaging of neuroinflammation in relation to MS, and I invite you to find these for more detailed information on this subject. So take home messages. There is increased TSPO binding in progressive MS compared to relapsing remitting MS and controls. The TSPO PET measurable innate immune cell activation predicts later progression in MS. And this gives proof of concept that TSPO PET measurable innate immune cell activation is detrimental for MS patients. And TSPO PET can be used longitudinally to quantitate change in microglial activation in therapeutic settings. And this has implications for the use of TSPO PET in treatment trials of progressive MS. But we definitely still need new radioligands with better specificity and better imaging properties. And with this, I thank you all for your attention, and I'm happy to take questions.
All right. Thank you for your very exciting presentation. I think this is an important growing field that teaches us a lot more information about MS. So just like before in the previous talk, we invited someone to start the discussion with you. And in this case, it's Martijn Wijberg, who is a neurologist in training here at our hospital, who also did a PhD in MS and actually on natalizumab in MS. So we're very curious what he will ask you. So Martijn, I think you're still muted, but just waiting for that. Yeah,
right.
Go ahead.
Yeah, well, thanks, Dr. Airas, for this really interesting talk. And also congratulations on the brain paper, which really convinced me that TSPO PET has a real potential to play a significant role in the future of MS treatment trials and perhaps even patient management for progressive MS. I'm just wondering, do you think that this technique, maybe it may be possible to use this technique even earlier in the disease course as for even treatment guidance, as you've shown that natalizumab is able to affect this microglial activation. And also, I think you've referenced it in the presentation that, for instance, that CIS patients who convert to clinical definite MS already show a higher TSPO uptake. So do you think you can use it perhaps even earlier?
Yes, so I think it's quite clear from the clinical studies that the more advanced the disease is, the more microglial activation there is. And really, like at group level, we can really see the differences only between the progressive patients and healthy control. So if at group level, you're looking at relapsing remitting patients, the differences are not really significant. But I think in individual patients, definitely. So if you could, for example, detect the chronic active lesions, which seems to be a bad prognostic sign, like in MS and already in earlier stages of MS. So that would be very, very, very helpful if we could detect those in our patients in vivo. But of course, we are lucky to be able to use or apply PET imaging for the study of MS. But as you know, and I know Amsterdam is also lucky to have it, but many places don't have it. And I guess now there are other methodologies emerging that could be also used for showing this pro-inflammatory microglial rim at the edge of the chronic active lesions, such as some MRI sequences like the QSM MRI, for example. So I think this might be something that could be then more widely available. And then, of course, what the field is also trying to develop very much is to create some biomarkers that could be reporting on the activation status of the central nervous system cells, and this would be really helpful also if you could use the biomarkers that you could measure in blood instead of performing actual PET imaging. So I think PET imaging is a great research tool. And I strongly believe that it can be also used in clinical trial settings, especially like maybe in earlier phases of the compound development. But of course, to really use it very widely, that's a challenge.
Right. Well, thank you. I see that there are many questions in the chat, Menno.
Yeah,
Shall I leave them to you?
Great, thank you. Thank you very much. So we have an interesting question from the audience. The question, I'll just read it out loud. It says, I am myself actually an RRMS patient, and I was wondering would it be a future treatment option to combine drugs that modulate both the peripheral immune system in combination with drugs that target the smoldering fires, so the active lesions directly, based on the TSPO PET outlook?
Yes, absolutely. I strongly believe that that will be the way in the future. So I think it will still be beneficial to block the trafficking of the peripheral immune cells into the central nervous system. But in addition to that, we need something that will directly target the pathology ongoing in the central nervous system. And I think one of the best targets at the moment is activated microglial cells. So definitely a combination treatment will come in the future.
Yeah, great. One more from Obada Alsgul. I'm sorry if I mispronounced your name. Thank you for this nice overview of TSPO PET imaging. I was wondering what your opinion is on the new SMW139 PET tracer, which targets P2X7 purogenic receptor. Do you think that this has the potential to replace TSPO? Very specific question, but
yes. Okay. So this is a very interesting PET ligand, and I guess the hope is that the target would be more specific, sort of in terms of detecting pro-inflammatory microglia, but I think we're still in the early days in testing this radioligand. But future will tell. But the thing is that with every new developed ligand, we just have to do the basic work. First we have to test just how it works in the human in general, and then we have to test how it performs in MS brain, and it all takes time. But I think this is definitely a very promising ligand.
Yeah. I have one final question that I noticed, which is, I think, related to the first question by Annemarie van Dam. What do you think triggers microglial activation in SPMS when lymphocyte influx is reduced, or also which also happens after natalizumab treatment?
Yeah, I mean, this is $1 million question. What triggers microglial activation? What keeps it up? I think this would be so useful to know, but I think to some extent there will be still trafficking, but at the lower level, also during the progressive stages of the disease. So so maybe even if we don't see the real pronounced gadolinium enhancement, but there may be still some damage of the blood-brain barrier and some lymphocyte infiltration through, and this slow or lower level trafficking is probably something that will keep up the microglial activation. But I'm sure there are many other factors that could be maybe modified or modulated in order to reduce the damage.
Yeah, I think it's an interesting field with a lot of movement also with regard to treatment. So very interesting. Absolutely. Thank you. So I think we don't have time for more questions, but we have a few more interactive things. So I think it would be nice to move to the polls now. So we have one more poll, both for Professor Airas and Professor Van Der Hoevel. And can we move to the first poll that we have? So after Martijn Van Der Heuvel's talk, hopefully you grasp the complexity of the brain network. And do you think that the complexity has a direct implication for our understanding of neurology or psychiatry? Okay. Okay. Well,
I only have to convince 2 people.
There you go. There you go. Almost there. Almost there. So what do you think?
Great to hear. It's good to hear that.
How can we convince them? That's the question.
Well, I'm not sure whether we need to convince them. I think we have to show that. I think that's. I was intrigued by Linda's question, saying, in the end, we have to show that it has clinical applications, and then I'm sure that the two people will start rethinking. But that's the big important step, right? That we show evidence that it is indeed.
Let the data guide us, basically.
Yeah, absolutely.
Right. Thank you. Moving on to the next poll. Do you think, back to Professor Airas again, do you think it would be important to take into account microglial activation when planning a treatment trial for progressive MS? So has she convinced you that this is now important? Aha. So Professor Airas, one person said no, and I think you convinced almost everybody. So that's good news. Thank you everyone for attending our first TN2 virtual meeting. And as I said before, if you like this type of approach, we have many more planned. These are the 4 that are upcoming. The next one will be on January 21st, and the setup will be the same. So you can ask lots of questions, and hopefully we'll have a more interactive session as well. So that was basically it. For the first edition, I would really like to thank Sabira, who really did a lot of planning for this work. And of course, our 2 speakers, Professor Airas and Professor Van Der Hoevel. Thank you so much for taking the time to be with us here today and to share your ideas and knowledge. And hopefully we stimulated some translational thinking in the field of neuroscience. So thank you very much.
Thank you.
Thank you very much for the invitation, and good luck for your translational neuroscience network program also in the future.
Thank you. Thank you very much. And hope to see you in Holland sometime soon after this.
Absolutely. I'm a big fan of Amsterdam neuroscience. You're doing great work here.
Great. Thank you. Okay. Thank you very much. We're at the end of the program. So see you in January. Thank you, everyone.